Still audible? Awesome. Yeah, after Shiny, Mark, Lohan, Shiny, and apologies to our friends from the West, I know how it feels to be like KL Rahul in the Gautam Gambhir era. I will nevertheless persevere. So, I have to work with what I get. So I'm again thankful for Mark and Shiny to introduce extracellular matrix in their respective talks. So again, but to repeat, extracellular matrix is all that surrounds cells, normal, cancers, senescent, non-senescent, and so on. And what we'll look at in the next 10 minutes would be some observations of how extracellular matrix confounds and interests the interactions between senescent cells, non-senescent cells, cancer cells, and so on. This galaxy happens to be my postdoctoral lab about 10 years back when my supervisor was invited to write a review on cancer in general. And seniors like Mark sitting here had almost said whatever was needed to be said at that time on what were the current interesting aspects to be spoken about the cancer microenvironment. So foolishly, I ventured in and I went in and we wrote this review, and I think when I read it now, I feel so foolish about doing that. But the title still seems to have a very nice echo, and that is Dialectics of Microenvironment. I was young, apologies, of the microenvironment and macroenvironment on the organ phenotype. So there was the microenvironment, the tissue, the macroenvironment, in which we would consider aging, senescence, and other such environmental factors. And akin to what Prashant mentioned at the inauguration, we were, the way I envisaged all these cues to sort of start playing on the tissue state, on homeostasis, was through some fanciful systems architecture-based narrative. In time and with wisdom, of course, we have a sense of the laboratory research phenotype, and our laboratory focuses on the early migration of cancer cells from the primary focus. We established how cancer cells are able to migrate through fluid environment. So we focused purely on cancer biology as a young group at Bangalore. When then came a microenvironmental influence, and there was Deepak with his laboratory just next door, talking about aging and senescence and so on. And then there was also a macroenvironment, and that was the COVID times. So that allowed us to ponder on what we were doing, and then take some brave new directions towards being able to focus the cancer research. And I started thinking as part of my group on trying to understand how senescence interacts with cancer. And our focus was always on extracellular matrix and how that regulates kinetics of cancer progression while it moves out, while it seeks new environments, and so on. So two stories on two interesting aspects or two interesting theaters of cancer metastasis. The first one would be introversation or movement of cancer cells into circulatory environments. The other one being colonization, or reaching the cancer, allowing the cancer to reach and decide where to habitate and eventually grow. Someone mentioned somebody works with one cancer, somebody with another. We work with two cancers. So anyway, the two stories and two cancers. This one. Colonization, the first story. What determines colonization of cancers in new stromal locales? How does the macroenvironment come into play? So, okay, clearly I need to move faster. So, what happens in ovarian cancer, this was work done by Bharat, who's right now a graduate student at Vanderbilt. What happens in ovarian cancer is, as ovarian cancers come and sit on what is called the serosal layer on top of the intestines, which is like the boundary of a circulatory cavity, they start spreading and moving and start growing around the normal cells that surround them. The normal cells, in turn, try to stall the growth of the cancer cell. So there's this intercellular competition between cancer cells and stromal, what are called mesothelial cells, that fight with each other and sort of bring about a defense-offense strategy at place. What we saw very early on using co-culture studies on mice peritonia ex vivo was that if these cells, if these mesothelial cells, if these normal cells were young, for young mice, or they were unsenesced mesothelial cells, they would be able to hold on and prevent the proliferation and migration of cancer cells. But if the cancer cells were to be co-cultured in, I'm trying to see if I can get the time lapse running on the left-hand side. Oh, there we go. But if the cancer cells were to be present amidst senescent mesothelial cells, irradiated mouse cells, aged mouse cells, they would start spreading much faster and they would actually breach that defense. So the offense of the cancer cells is far better in senescent peritoneal microenvironments. So we then asked, what is happening here? And we found that one of the earliest cell biological observations we made was that ovarian cancer cells preferentially tend to attach closer to senescent mesothelia, and they tend to extrude these senescent mesothelia far more. We also then resorted to computational multi-scale modeling, where we asked what should be the minimal set of criteria between normal cells, cancer cells, normal mesothelia, and senescent mesothelia. So the idea being that in an aged environment, not necessarily all cells tend to be senescent cells. So within such computer modeling, what we also found out was that the cancer cells also needed to adhere far better in such environments. They tend to adhere better in senescent microenvironments. So what allows cancer cells to better attach and then remove the normal cells in this environment, enhancing their competition? And that turned out to be, again, the matrix. So we found out that senescent cells, senescent mesothelial cells, senescent normal cells, tend to secrete a completely different composition of extracellular matrix, something that compositionally is glycan-rich, that allows cancer cells to better attach and start better spreading. So you can see the exploration of the cancer cells on senescent matrices is far better than that of cancer cells on non-senescent matrices. I'll come to the take-home message one more time, since I have to move on to the next story. The next story goes into the question of what determines introversation of cancers. Now, any kind of process that involves breach of cells from one tissue to another tissue, in this case, the cancer cells from their site of origin to a new environment, in this case a fluid environment or a blood environment, needs some snazzy bioengineering. And Shiny, again, was able to talk about these organ on chips. So we took that particular route, and Nilesh, who's a graduate student who's about to graduate from the lab, worked on this very interesting sort of horizontal multi-organ on chip setup, where he was able to bring in a tumor environment and rig it up in continuum with a vascular-like environment. So we created a blood vessel and a tumor environment, all in a horizontal setup, such that we can view everything together under a microscope in real time and watch the entire process of cancer spread or cancer entry from the cancer environment into the blood vessel, perhaps even collect the cancer cells at the end, and so on and so forth. Those are the advantages with such setups. In such setups, green being the endothelial cells, oh, goodness gracious, I'm getting confused between the two things that have been given. Green being the endothelial cells, red being the cancer cells, we were able to see how the cancer cells and the endothelial cells were able to come together and form anastomosis, with which allows the cancer cells now to move from the stromal compartment into the vascular environment, which was a microfluidic environment in which we were able to flow blood-like fluids at a constant rate. And so the cancer cells were able to breach and actually move in, using all the interesting histological features associated with such processes. Now comes the macroenvironment once again. So the macroenvironment in this case was this particular metabolite called methylglyoxal, which is an unintended waste product of glycolytic flux. Glycolytic flux tends to increase in certain stresses, which includes metabolic stress, such as in diabetes or even in aging. This particular metabolite, in fact, again, doff to Mark's comment on the multiple hit hypothesis, this particular metabolite is able to actually create that second hit in the presence of one BRCA hit in breast cancer, as has been shown by Ashok Venkatraman's group in NUS, and it is able to bring about cancer progression by itself. So it acts like a oncometabolite on some level. So we started flowing this metabolite within such environments to see how introversation kinetics can now be exacerbated by microenvironment and the macroenvironment, the diabetic, aged, metabolite macroenvironment coming together. We saw that there was greater efficiency of introversation. Cancer cells were able to breach this far better. And how were they able to do that? Remember, the breaching of cancer cells into the fluid requires not just their ability to breach the endothelial cells, the cells that make up the blood vessel, but also all the matrices that they encounter from the start till the end. This includes a stromal matrix, which they destroyed by hyperpolymerization, the endothelial matrix, which they dissolved as well as they moved through, and they were also able to break through the endothelial cells by making them senescent. So the methylglyoxal, the metabolite, is able to denature the matrices in unique ways, allowing for the cancer cells now to move more smoothly into the fluid environment. The cancer cells also lose their adhesivity onto such matrices, but cancer cells, upon losing adhesion, behave like amoeboidal cells, and like amoebae, they are able to worm through these larger spaces at much faster rates, explaining perhaps the introversation kinetics. So, take-home messages. Introversation, or movement of cancer cells into circulatory compartments, into blood-like environments, involves harnessing macroenvironmental cues such as the diabetic metabolite, such as methylglyoxal, which allows it to hit several different components along its way. Second take-home message, in case of colonization, the winning of cancer cells over the natural defense barriers that are there is brought about by allowing the senescent mesothelial cells to make unique matrices that act as better homing areas for the cancer cells to attach. In other words, in senescent environments, cancer cells have better adhesion, which allows them to attach and extrude the normal cells, therefore allowing for better cancer invasion. I have just a plug for my students who have posters today evening. Greater details of the story that I talked about is by Bidita. Anchita and Inbasekar also have posters on two very interesting aspects of how senescence and cancer collide, and also ways by which we can perhaps even control such processes. Thanks again, of course, to the students of the research group that I manage, to collaborators who brought in great ideas and tools and concepts to the table, as well as to funding agencies. And I'll be happy to take questions. Thank you so much.
Thank you, Ramray, for that vibrant talk. The floor is open for questions. And Lawrence first. A mic there, please.
Thank you very much for your talk. I really loved it. And of course, I have two questions. First question on the homing of the cancers to the senescent cells.
Yes.
I was wondering whether there are two stuff. First, they are able to detect and to get there, and then they can adhere better and do their job.
Yes.
So could it be that the senescent cells, they have a much lower metabolism, they don't use as much fuel, glucose, oxygen, and so on, and then it would kind of select the cancer cells to be better and more active in the sub-microenvironment of the microenvironment where the cells are senescent?
Right. So we did look at the diffusive or the secretory parts of the senescent cells, the whole SAS pipedea. We saw that was not playing much of a role here. Rather, both the modeling as well as the experiments indicated that things were more happening in a more local way. The senescent cells are making their substrata around them more stickier, and that's what causes the cancer cells to home in. Subsequently, though, there could be CXCR, CXCL driven chemoattractant mechanisms that might bring the cancer cells closer to the senescent cells. But the stickiness driven by hyaluronic acid, that's the matrix that's there, is what brings the cancer cells closer to senescent cells.
Second question on the glycation that you showed us.
Yes.
That's quite fascinating because glycation is used in patients to look at in diabetes and to look at this kind of indicator. And it is also states that are correlated with a higher incidence of cancer metastasis. Do you have epidemiological evidence?
Yes. Methylglyoxal is a dicarbonyl. It has one aldehyde, one ketone. Far better affinity for protein lysines than does glucose.
Thank you so much, Ramray, for that wonderful, vibrant, and
you.
multidimensional talk.